All right, it's a pleasure to introduce Russ Poldrack from Stanford, who has given a version of this talk every NeuroHackademy so far. And I'm sure you will enjoy it and find it thought-provoking. Thanks for being here every time, Russ, and take it away.
Yeah, thanks for having me. All right, let me just screen. All right, does that look okay on Zoom now? Cool. All right, well, nice to see you, meet you all. Just minimize that. Yeah, so I want to, my goal here is really to talk about why we think, caring about issues around reproducibility is an important thing. Actually, let me just grab my little and I would have, in some ways, I guess I would have hoped that I shouldn't talk. I might have hoped that, how many years has it been now? This is the seventh, eighth, right? After 8 years that I wouldn't have to be giving this talk anymore, but the issues that I'm going to raise are still fresh, I think. So hopefully it'll, convince you that this is important. Let's see. Let me just move that down here. Is there a way to make this thing go away? I guess not. No.
Not reliable.
Yeah, right. Okay. Well, we'll kind of stick it down here. Hopefully it'll stay out of the way. So if you go back, this is going a couple years back, go to NIH and look at, how many grants mention functional MRI in their abstracts, and how much money went to those grants. Over 1,200 grants said something about fMRI, and those in total had a cost of almost $500 million, more than $500 million, $552 million. And if you take all the abstracts and do like a word cloud on them, just to try to get a feel for what people think fMRI can tell us, these are the words that come out: brain change, treatment, clinical, patient, mechanism, fMRI, behavioral development, individual. Clearly, people think that fMRI is going to be useful for telling us something about how mental illness works, how brains work more fundamentally, what the mechanisms are in the brain that give rise to thought. And so the question is, should we believe that? And how has it actually paid off? I mean, people, NIH has been funding fMRI research for 30-something years now, since fMRI started. So there's some kinds of questions that you might hope we could answer, like, what's going on in the brains of people diagnosed with depression? And a number of meta-analyses have come out over the last few years, suggesting that there were consistent findings in studies of depression using task functional MRI. These are some of the results from that. And Mueller et al., one of the world's top teams at neuroimaging meta-analysis, basically found out that there were some problems with these earlier meta-analyses. They reran the meta-analysis. This was over a total of 99 different studies. Each of those individual studies had said, we found a difference between people diagnosed with depression and people without a diagnosis doing some particular task in their brain, and told a story about why that was important for depression. But it turns out if you put the data together from all those 99 experiments, you basically get nothing that's consistent across them. That seems like a problem. We've also, seen cases where, yes.
How do you put the data together?
There's not going to be anything on coordinate-based meta-analysis, is there? So it's an approach called coordinate-based meta-analysis, where you basically take the activation locations that were reported in each study. People generally report these things in a way that's sort of consistent across papers, and you can think about it as like reconstituting the imaging data from the table that says where activation was found, and then do statistics across those. It's
the same task.
Or related tasks. Yeah. It's a pretty well-established approach. It has problems, but it is a pretty well-established approach. If there were something consistent, I mean, it works in a lot of cases. when things are consistent. So there's nothing consistent here, right? So, if fMRI and machine learning could identify people who are at risk of suicide, that would be a great thing, right? Suicide is a huge problem and a growing problem, here and elsewhere. And this paper published in 2017 claimed to have found that machine learning could be used to identify suicidal youth. They show them words related to suicide and non-suicidal things, and they show this brain activation pattern for the word death in participants who had made a suicide attempt compared to control participants. And they ended up, the investigators who published that paper a year later ended up getting a $3.8 million grant from NIMH to go use fMRI and machine learning to study suicide. That 2017 paper was retracted. This came out earlier this year after it became clear that really due to work by Tim Verstein and Conrad Herding digging into what these investigators had done, that they basically did the analyses wrong. They double dipped their data. And this is from the authors. They say, we confirm that their method was indeed flawed, and that the results don't hold up in their new analysis. So clearly, there are still problems in the fMRI literature that are leading to problems with reproducibility. Yeah, it's a mess. And so how do we fix it? I'm not going to spend much more time talking about the mess. I'm going to try to talk about how, we and others have tried to fix it over the last now more than a decade. And one of the sort of inspirational quotes, I guess, for, thinking about why we've done a lot of what we've done is this quote from Louis Brandeis, former Supreme Court Justice in the US. Sunlight is said to be the best disinfectant, right? The idea is that if we're doing things as transparently as possible, then you're more likely to find problems, right? Another version of that is Linus's law: with enough eyes, all bugs are shallow, right? Enough people looking at things, you're going to find all the problems. So I'm going to talk about various aspects of this sort of ecosystem that we and a bunch of others, some in the room, have been involved in building for a while. And it's really, I guess it has sort of two parts. One part is really the data sharing part. And I'll say a bit about Open Neuro, which is the platform that we've developed for data sharing. I'm not going to say a lot about it, actually. And then there's a whole other set of tools, a whole ecosystem that's really based around Python, that's ultimately led to the development of sort of full-blown workflows like fMRI prep and MRI QC. And there's a lot of stuff here that I think you'll be encountering throughout the next 2 weeks. A lot, especially the things at the bottom down here, which are really like the infrastructure that if you're doing anything with Python and neuroimaging, you're going to be working with that infrastructure. Things like NiBabble and NiPipe and BIDS. So I like to think about fixing science as kind of a design problem. Does anybody know what this is? Raise your hand if you have a guess what this thing on the screen is. Yeah. It's a paper cutter. And so, yeah, it's a commercial paper cutter. And why do you think it has 2 green buttons? Yeah.
Make sure no one can in the paper.
Exactly. People are going to have the tendency to hold the paper in place with their hand and cut one of their fingers off. So that basically the idea is when you design a system, you want to think about what are the ways that people might do things badly, and you want to design around that. And that's, really the idea of design in some sense, is like figuring out, building the right affordances into systems so that people will do the things you want them to do. And, we've known, I think as we started learning about all the reproducibility problems in science, people started talking about the degree to which, humans are really good at fooling ourselves. We're really good, for example, at seeing things that fit with what we believe and kind of not looking too hard at the things that don't fit with what we believe or what we want to see. We'll come back and talk about some specific examples of that as we go along. But another way to think about, this problem comes from behavioral economics. So there's an idea in behavioral economics called choice architecture, right? And the idea is like anytime you go into a situation, there are particular features of that situation that cause you to be more likely or less likely to make some particular choice, right? If the If the apples are sitting at the, at the checkout register at the grocery store, you're more likely to buy an apple than if they're way back in the corner, hidden behind a bunch of other stuff, right? And the idea of nudges that Thaler and Sunstein and others have talked about is really that we want to push, basically build systems that kind of drive people to do the things that we think are the best things to do. And if we're not, they still have the choice to do it a different way, but especially things like defaults, right? Anytime you use software, there's almost always going to be defaults, a default threshold for this or a default smoothing kernel for that. And to the degree that those are good defaults, that's a good thing. If they're bad defaults, you're going to basically be pushing people towards doing bad things. And we also need to think about how do we sort of expect what kinds of errors people are going to make and build systems to try to expect those and prevent those. How many of you have heard of this paper? Okay, so this is I think it's a paper that you all should go download and read. It was published in 2005, but it's about as, I don't think it's any less relevant now than it was then. There's arguments about whether you really believe all the claims that he made, but I think most of them, at least at some kind of general level, have been shown to be right. So he basically made the argument, John Ioannidis made the argument that most published research results are false. And I'll say more about the basis of that argument later, but he also in the paper, he went through and kind of talked about what are some of the factors of particular studies that might cause them to be more or less likely to be true. And the first thing he pointed out was that the smaller the studies conducted in a particular scientific field, the less likely the findings are to be true. Why would that be? Well, first, let's just point out that, in general, neuroscience research is pretty badly powered. So these are data, this thing isn't going. These are data from two different studies. Let's try it up here, maybe it'll be less annoying. From two different studies that have done sort of meta-analyses of sample sizes across different literatures. The one on the left from Kate Button and colleagues was focused on human structural imaging and rodent studies. And then the one on the right is really broad across a lot of different domains. And basically what they show is that the statistical power of most studies is really bad. So statistical power is the likelihood that if there is a particular effect there, your experiment will find it. Right. So if there's, let's say that you want to know, does smoking cause cancer, right? And you go do a study and you say, let's say you go do 100 studies and ask how many of them will are going to be able to find that effect of smoking causing cancer. Power tells you what that proportion will be. If your studies are well powered, which usually comes from having larger sample sizes, then you're going to be very likely to find that effect when it's there. If your studies are not well powered, which usually means from having very small sample sizes, then you're unlikely to find it, even if there's a true effect. And so in the limit, that means you're basically doing futile research, right? Nobody wants to do a study that even if the effect is there, the study can't find it, right? It's like doing going and doing astrophysics, but your detector is broken. We'll come back to that example, right? Actually now. So, what we learn about in statistics about power is just how likely are you to make what we call a type 2 error, right? Of basically saying that there's no effect when there really is. That's separate from controlling your false positive rate, right? How likely are you to say that there is an effect when there isn't one? And so some people have thought that means that, well, the likelihood of getting false findings doesn't depend on power. But what Ioannidis did was kind of framed it a different way, where he said, what we really care about is what we call the positive predictive value, which is instead of looking across all studies and asking what proportion of them are false, which is what our type 1 error does, looking only at the studies where we claim to have found a positive result and seeing how many of those are false. Actually, how many of them are true is the positive predictive value. How many are false would be what we call the false discovery rate. So it turns out that the likelihood of one of your findings being true, when you have a positive finding, the likelihood of it being true is related to your statistical power. So let's do a thought experiment. you're a physicist, you're trying to detect gravitational waves, right? But you're unbeknownst to you, your machine, the machine you're using is like broken, it's just giving you noise. And so you go do 100 experiments, and let's say you're fine with a 5% false positive rate, you're going to get 5 positive results out of those 100 experiments. They're all going to be false positives, right? Because your machine is broken, it's only giving you noise. If you go and like fix the machine, now the proportion of all the positive results that are true is going to go up because your machine's going to start finding the true things, assuming that there is a true thing to be found. And that's really the point that Ioannidis made. And so a main part of the argument about why most published research results are false is most published research results are underpowered and also have the other problems that we'll talk about in a bit. There's another issue that happens that we call the winner's curse, where when your sample size is small, the only effects you can find are really big effects. And so if you then go and ask, how big are the effects out in the literature? They're all going to be huge effects if the sample sizes are all small, even when the real effect is, the actual effect is much smaller because you're only able to find the really big ones. So we end up overestimating how big the effects are based on the literature. This is a great example of this that came from Felix Schombrot and Perugini a while ago, where they looked at this thing they call the corridor of stability. The idea is each of these lines, what they're doing is they're just sampling data from a distribution and looking at the correlation. And they're always sampling from the same distribution. So in theory, the true number here, the true correlation between X and Y is 0.225, I think, 26. And the idea is, let's start with the dark line is like one sample. We start with, I don't know, 5 data points and keep adding data points. And so that as the line kind of jumps up and down, that's what's happening as you add data points, as that single sample gets bigger and bigger and bigger, until it finally, at some point, approaches the true value, right? But this is after several hundred samples. So the idea is that early on, when the true correlation is 0.26, you could get if you have small samples, you can get a value anywhere from like 0.7 to minus 0.25. And it's not until you get past this, what they call the point of stability, which for their analysis was 161, that you start actually seeing things that are relatively close to the true answer. Many of you may have seen this particular paper that came out to a great deal of fanfare last year by Scott Marek and colleagues, which basically claimed that and showed that if you're analyzing brain behavior associations without very large samples, you can end up basically having very unstable effects. And in part, that's because most of the true effects, when you look at them in an unbiased way in a very large sample, are actually very small. These are the distributions of the true correlations between various brain measures, either connectivity measures or structural measures, and various behavioral measures. Sorry, these are tiny down here, but basically, if you go look at the paper, I think the most compelling thing they show is that if you take a bunch of samples of 25 people, what you can basically find is, this is the true correlation here for the full data set is minus 0.08. But if you take small samples from that big sample of size 25, you can find one sample with a negative 0.64 correlation, one sample with a positive 0.5 correlation. And so if you're just looking at very small samples, you're basically setting yourself up to be fooled by randomness. And this has happened to, like, there was an entire field that we now think, at least there's probably multiple, but there's one entire field that we think was largely people being fooled by randomness, which was the study of what were called candidate gene association. So back around 2000, it became possible to relatively inexpensively measure which particular variants, which versions of a particular genetic polymorphism a person had. And so people started publishing studies saying, hey, this particular variant, whether you have the VAL versus the MET version of BDNF, is associated with how big your hippocampus is in this example. And there are a lot of other examples of this. this is from a meta-analysis that looked across the literature over time. So what we're plotting here is on the Y, what they're plotting here is on the Y effect is the effect size that was reported in the paper, right? And so these are like, effect sizes of 1 are, we think that anything above like 0.8 is a very big effect, right? So these are like whoppingly large effects of supposedly of a single difference in your genome out of the 2.2 billion places in your genome where you could differ. This is time, year of publication on the x-axis, and the size of the bubble is related to sample size. So what do you see? You see over time, sample sizes get bigger. Early on, there were some very small sample studies with reporting very large effect sizes, right? Nothing being reported down here, note that. Over time, it tends towards 0. And in fact, we now know that that effect is 0 because people have done very large genome-wide association studies where you have tens of thousands of people in the study. And this is a quote from Jason Stein's paper. In general, previously identified polymorphisms associated with hippocampal volume showed little association. That includes BDNF along with a bunch of others. They did find other genetic variants that were associated with hippocampal volume, but it was none of the ones that people had thought of. And so now, if in the genetics literature, you can no longer publish a small sample candidate gene study. Unfortunately, you still can in the cognitive neuroscience literature, and you see them occasionally. So how well powered are fMRI studies? How bad is our problem? So it's hard to know. You have to, without going in and like reading each paper and saying this paper had, N of 25, which is a labor-intensive thing, given that there's, thousands of papers being published each year at this point. You have to make some guesses. And so there's various ways. So this plots on the left side sample size per group. So if there's like multiple groups, this would basically be looking at the size of each group, not the total size, because we think that that's what matters for most of our comparisons. Sean David and his colleagues actually did some manual annotation of early papers. That's the lighter blue line here. And these are various analyses. The PubMed abstract one is basically done by taking all the PubMed abstracts that mention fMRI and seeing if Talia Honey wrote some code a while ago to basically heuristically pull out sample sizes. And so we get not all the abstracts mention it, but some of them do. The green line here is from a similar analysis done on full publications from PubMed Central by Kendra Udaik and Jerome Dacus from MNI. And they roughly tell you the same thing, right? Sample sizes have been going up. And they're, now they're roughly, so in 2020, the median study was an N of 35 per group, which is certainly better than, it's been going up. And I think a lot of it is people are starting to use these very large data sets like ABCD and Human Connectome Project. Now, what we want to know is assuming that your sample size is that big, how big is the effect that you can find, right? When we think about power, we want to have sufficient power to find a reasonable effect size. If I tell you I'm going to build a system that is really good at finding differences in height between people, and then you ask me, well, how big of a difference can it find? I say, well, it's really good at detecting the difference between a 2-year-old and a 20-year-old, right? That's not a very good system, right? Whereas if my system can detect the difference between a 13-year-old and a 14-year-old, then which is a much smaller difference, then we might think it's a better tool. So if we look at, we have to make some assumptions about what we're trying to find, and this is based on some previous work that Yoka Durnas did in my group. We find that the median study was powered to find an activation with a effect size of 0.67 or so. And the question is that a plausible effect size for, if we're looking for interesting effects in neuroimaging data, should we expect those interesting effects to have an effect size of around 0.7? Now, the problem is you can't take the published effect sizes from the literature because of that winner's curse thing I told you, because we're looking across hundreds of thousands of voxels and we're saying, where is their activation? And then we're looking at the effect size after having done that thresholding. So we're already taking the winners. So what we have to do is find a way to estimate effect sizes in an unbiased way. So what we did, this is from our 2017 Nature Reviews Neuroscience paper. We took data from the Human Connectome Project and did this for a number of different task fMRI paradigms. We got we took their data, got a group map, then we needed to know, where do we look to get an unbiased region of interest to then measure what the effect size is. So fortunately, the Human Connectome Project group had actually written down regions of interest. And then we can also look at neurosynth, which I'm sure you'll hear about at some point, which is another way of doing this sort of like coordinate-based meta-analysis. And we got regions where we're pretty sure that, for example, for a motor task, there should be activation. And then we put those all together to give us an unbiased effect size. So this is a plot of those effect sizes. So there's two things plotted here. So in task fMRI, what we sometimes measure is like how much for one person, if I show you, say, a bunch of faces and I look in your face area, how much does the signal go up in terms of like how many percent does it change? We call that percent signal change. That's a measure of like the activation within a person. And for these are, for some of these tasks, it gets up to about 4%. That's about the biggest you'd ever expect a signal change to be. Like you'll sometimes see signal changes on the order of, say, 4 or 5% in like primary cortices, like primary motor, primary visual. That's whoppingly big. Like interesting effects are usually much smaller. Even for like, gambling task and emotion tasks, you're getting like around 2% signal change. So these are already like really big, but that's not the effect size we care about, right? Because when we're doing statistics, we're asking how does the mean, how likely is the mean different from 0 across subjects? So we have to look at the distribution across people. So we take that, the measure for each person, and then we ask how far is that from 0? That's this Cohen's d that we're computing. And I mentioned, something like 0.8 is a large effect, 0.6 is 0.5 is a medium effect, 0.2 is a small effect. And what you see is that, most of these are like, basically, even though they're whoppingly big, in terms of like how big the signal goes up, the effect sizes are actually not that huge, right? Some of them are huge, right? Like motor cortex turning on when you do a motor task, that's about the biggest effect you're ever going to see. And that's a Cohen's d of about 0.7 or so. So what this is saying is that the median fMRI study in 2022 was barely powered to find the whoppingly biggest fMRI activation that you could expect, right? Most interesting fMRI activations are probably an order of magnitude smaller. So we're still badly underpowered. If we go back to look at that Mueller et al. Meta-analysis, this is plotting data. Each point here is one of the groups from one of those samples, plotted over time and plotted as a function of sample size. And I've colored the ones with a group size lower than 20 in red. And I do that based on this claim. Simmons et al. Wrote this paper called False Positive Psychology, which is a great paper. It's the paper that coined the term p-hacking. And they had a bunch of suggestions about how do we make science better. And one of them was authors must collect at least 20 observations per cell or provide a compelling justification for not doing so. And the argument is that samples smaller than 20 per cell are simply not powerful enough to detect most effects. So there's usually no good reason to decide in advance to do that, right? In general, what it probably reflects is people collecting some data, doing sort of, interim analysis, finding a significant effect, and then stopping, what we call flexible stopping, flexible termination. And we think that a large part of why that literature is so irreproducible is probably because of the low power. Now, it isn't any better in machine learning. You might hope that the magic of machine learning would save us, but it turns out that small samples in machine learning just give you much more variable estimates of predictive accuracy. This is work from a great paper by Gail Veracruz called Cross-Validation Failure that laid out, based on several different sources of data, the degree of variability in predictive accuracy estimates that are related to sample size. That follows basically what you would kind of expect just by a binomial rule. But maybe more distressingly or importantly, he looked at literatures where people have reported and done meta-analyses of predictive accuracy. And in almost all of these, you see, just as we saw earlier, reported accuracy goes down as a function of sample size. Right? Why is that? Well, it's probably because, it's a combination of there being a lot more variability when you have small sample sizes. And then if you have publication bias that drives people to not publish, null results, you have any kind of analytic flexibility that lets people try a lot of things until they find the best result. That's going to result in exactly this thing. It's come to be called the decline effect. It's kind of a general phenomenon that you see across lots of areas of science. Yeah. Sorry, each dot represents a study. And so it's basically plotting sample size versus the predictive accuracy reported in a particular paper. So each of these is from like meta-analyses across a bunch of different studies. Okay, so now, when I go talk about this, people come up to me, you've probably had this experience, right, where you have a visiting speaker and students go to lunch with them and you have conversations about, what they talked about in their talk. And what a lot of people say is, hey, how can I, I'm a grad student, my PI doesn't have the money to go collect a huge sample. How can I get a dissertation that I can, believe in and be proud of when we don't have the resources to deal with all the issues that you just brought up here. So I actually wrote this paper called The Cost of Reproducibility a few years ago that tries to lay out at least a few of what I see as kind of ways to deal with this. And one, I think most generally, the quote from Nancy Canwisher kind of says it all, right? If you can't answer the question you love the question you can. Because you're not always going to be able to do what you want to do, in graduate school. So you should find something you can do well and do that thing. And so some of the pivots that I talk about are one, collaborating, seeing, you may not have enough data on your own, but if there are 3 other groups that have data that you can bring together, that's what the genetics world has done now. Now, if you look at the genetics papers, they have hundreds of authors because they're often data from many different groups sort of put together. Using shared data, there's a ton of shared data out there, and I'm happy to talk a lot more about that. Another is focus more on theory or on computational modeling or analysis questions. Now we are in a kind of a golden age of data sharing, right? Just in the last decade, all of these, projects have come about sharing really amazing data sets from, really, well-known big ones like the HCP, the Human Connectome Project, and the Adolescent Brain Cognitive Development, the Biobank, which is a monstrous data set. We run Open Neuro, which has, over 800 data from over 800 different studies, over 30,000 people. If you're not familiar with the Natural Scenes data set, it's a really amazing data set for people who want to do vision science or memory research, probably lots of other stuff as well. So I think that, there's the resources to do analyses on shared data are getting better and better. And so I think that's one great way to think about going. All right, so what else did Johan need to say? The greater the flexibility in designs, definitions, outcomes, and analytical modes in a scientific field, the less likely the findings are to be true. This is a plot from that 2017 paper where we went through, this is just for the FSL software. We went through and asked, how many plausible different analysis workflows could one come up with just using that software? Just using like reasonable options at each step. And it turns out that the answer was like almost 70,000 different workflows. That's just for one software package. So obviously there's many more for others. And it turns out that people actually use those. So Josh Karp had a great paper he called The Secret Lives of Experiments, where he looked at what people reported using and showed that, data collection analysis methods are highly flexible with nearly as many unique analysis pipelines as there were studies, 241. People were just all over the place in terms of how they did everything. And it matters. So Josh also had another paper that took one of the data sets from Open Neuro and analyzed it using over 8,000, I think it was over 8,000 workflows. And he looked at the range of the statistics from highest to lowest. First he looked at the mean, and the mean looked like, it was a data set from our lab. Fortunately, the mean looked like what we had published. But if you look at the range, there are voxels where, depending on which workflow you choose, there can be 8 standard deviations difference from the highest to the lowest value, right? That's a monster difference in statistics. And so, the workflow matters. Again, machine learning doesn't make it better, it probably makes it worse. This is a great paper from Brad Weibel's group called "I Tried a Bunch of Things," which is basically about how you can very easily, get overfitting using, if you basically rerun cross-validation of the same data, you can end up finding what look like, really good results when there's really just noise in the data, basically just by tweaking your hyperparameters a little bit. It's also not just fMRI. This is a great paper by Luck and Gasplin, which showed that you could do things that people commonly do in the ERP literature and get bogus effects more than half the time. Okay, so how can we start to fix these problems? So one, now pre-registration, how many of you are familiar with the idea of pre-registration? Okay, good, most of you are. For those of you who aren't, the idea is you want to basically write down what you're planning to do in a place where it's sort of like held in an immutable form. So I put it into something like this place called the Open Science Framework. It's dated so that now I can go do my thing. And I said, how many subjects am I going to collect? What's my exclusion criterion going to be? How am I going to analyze the data? All this sort of stuff. So the idea is that this then prevents you from like presenting results that are really exploratory as having been predicted, right? It doesn't mean that you can't show your exploratory results. It just means that you can't present them as having doing what we call harking, hypothesizing after the results are known, like pretending like you predicted something when you didn't really predict it. One of the problems with doing pre-registration in the context of imaging is that it's really hard, right? You're not just doing one test. You have this complicated workflow. You have a complicated analysis stream. And so I think people often have the idea that, well, if I pre-registered it, then I can't do anything differently. And that's just not the case. I mean, every time we've published a paper having pre-registered it, we end up with a section called deviations from pre-registration, where we say basically, hey, we had pre-registered this, we ended up doing it this way for this reason. And if you have a reasonable reason, it's never been a problem. I mean, some reviewers get really upset if anything changes. But if the pre-registration says that you have to do something that you now realize is a bad idea, you shouldn't go do that thing just because the pre-registration says so. You should say why you didn't do it that way and why it's a bad idea. And you can even do this with existing data. Obviously people have to trust you that you haven't actually looked at the data, but if we can't trust each other, then we might as well quit, right? So this is a pretty amazing demonstration of the effect of pre-registration in another domain. This is in clinical trials for treatments of studies funded by the National Heart, Lung, and Blood Institute in the US. They're funding like clinical trials on things to prevent strokes and heart attacks and stuff like that. So they put in a requirement in the year 2000 that any clinical trials had to register their trial on this site called clinicaltrials.gov. And they had to say, what's the primary outcome, right? What's the thing that you are going to be measuring that you're going to count as success, right? Is it all-cause mortality? Is it number of strokes? Is it, reduction in some particular measure? What is it? Before that requirement, so the things with a plus sign in them are clinical trials that claim to have found a benefit of the treatment. The empty ones are ones that claim to find no benefit. There weren't any before 2000 that claimed to find harm of the product. Fast forward after 2000, nearly every trial fails to find a benefit or a harm. One finds harm, two finds benefits. It's not as if drugs stopped working in the year 2000, right? What happened is people running clinical trials could no longer move the goalpost and make it look like by picking a primary outcome that they could say, hey, there's an effect on. They had to actually use the one that they had planned, and suddenly they don't get the kind of effects they used to get. I think it's a great demonstration of how it really can kind of prevent us from fooling ourselves. Now, this doesn't eliminate analytic variability, right? My lab would pre-register one pipeline, your lab would pre-register another pipeline, and then the question becomes, how big of a difference does that make? And so we wanted to know about like how much does this matter in the wild? Like you go to real fMRI labs and have them analyze a real data set, how much will it matter? So we did this study in conjunction with, it was really run by Tom Schoenberg and Rotem Nazaire, and then in collaboration with a bunch of economists, to ask what happens when you do this. So we collected a data set. Tom's group collected it at Tel Aviv, 108 subjects doing one of two versions, similar versions of a gambling task that we had run before in my lab. I'm not going to go into the details. I'm happy to talk more about it later. And then what we did was we tried to get people to take the data and analyze them. And we had a set of hypotheses we wanted them to test. We recruited people on social media at conferences. Each team could have up to 3 people. They had to sign a non-disclosure agreement basically saying they wouldn't tell others what they found until after the embargo was lifted. And all of them ended up being offered co-authorship on the initial paper, which is why the paper has about 200 authors on it. We gave out the raw data set and we gave them about 3 months to come back with their results. And the primary results were 9 questions where we basically said, is there activation in some particular brain area for some particular part of the task? Is there activation in the ventromedial prefrontal cortex when the amount you could win on the gambling task goes up, for example? And we basically said, analyze these data as if it was a study in your lab. The only restriction is it had to be a whole brain analysis. I think we gave out the data to 82 teams and we got results back from 70, which was pretty amazing. And we also got a bunch of other information back from them, like their maps before thresholding and after thresholding, and a bunch of information about their design. And we had them report their designs in a pretty systematic way using this thing called the COBIDAS standard for fMRI reporting. That even wasn't enough. Like we often still couldn't tell what they had done. And in some cases, we had to go ask them for code or for files to really figure out what they had done. But it turned out that none of those 70 teams, no 2 of those 70 teams used exactly the same analysis workflow. So what's the effect? So what are we looking at here? We're looking at the y-axis here is what proportion of teams said yes to this hypothesis. If everybody agreed, these numbers would all be either 0 or 1. Either 0 would be everybody saying, no, there's no activation. 1 would be everybody saying, yes, there is activation. Clearly, they're not all 0 or 1. For 4 of them, it's pretty good, right? These 3, pretty much everybody agrees there's nothing. This one, pretty much everybody, 85% of people agree that there is something. But then for these 5 hypotheses, like, is there activity in the ventral striatum for when the size of the gain goes up? 20 to 40% of teams are saying yes. So there's across the 70 teams, there are 33 different patterns of outcomes. And for any hypothesis, there's at least 4 workflows that can give you a yes answer. That seems problematic. And if you look, this is looking at basically, if you go voxel by voxel and say, how many teams are saying yes in that voxel? The voxel with the maximum overlap, which I think was like over here in like motor cortex for this particular hypothesis, had 76% of teams. So the best we did at any particular voxel was 76% of the teams saying, yes, there's activation here. This one is pretty amazing. For all this is across the 9 hypotheses. You look across the different teams, there's always at least one team that says there's no significant voxels at all. The minimum is always 0. And the maximum goes up to like, more than, there's like 220,000 voxels in the brain for this atlas. So basically there's some teams saying that more than half the brain is active. So none or more than half. Seems like we should be able to decide between those 2 a little more easily. So we dug in a little more deeply to try to figure out what was going on. And it turns out that, so this is a correlation map between the underlying whole brain statistics. And this big kind of, red blob up here says, well, there's a lot of teams whose maps are actually pretty close to one another. They're not that different from one another, even though, like, you can get teams whose maps are very highly correlated, but the red here means that this team said no, green means this team said yes. So something's happening between having really similar maps across the whole brain to saying yes or no. It might be, what do you consider ventromedial prefrontal cortex to be? Because we didn't give them an official definition. It might be, how did you do thresholding? There's lots of things it can be. Then what's going on here? There's this set of, 8 teams who are anti-correlated. They're systematically reversed from the other teams. It turns out that they just did the analysis wrong. It was a complex analysis, and their map ends up looking like kind of the whole brain activation map because of the way that they modeled things. And it turns out that the actual map for these these reward or sort of like, monetary gain contrasts ends up looking more like the default mode network, which is negatively correlated with whole brain activation. And that's why they end up being negatively correlated. And then there's a number of teams out here who are just kind of off in the wilderness, not clear what they were doing. So the point is that, we're not doing terribly, as long as you do the analysis right. But getting from the statistical maps to those decisions is still a really messy process. So what we could also do is just take all the results. This includes all the teams that did it wrong, and say, let's do a meta-analysis. Let's combine all those 70 maps and say, what do we find? And it turns out when we do that, we can find consistent results that look like, roughly what we would expect, at least in the places that we know about. So it's not as if the data are all garbage, right? There is consistent stuff going on. It's just that you have to look across lots of different analysis workflows to see it. And so this has really driven us to think a lot more about how can we go beyond the idea like there is a single workflow and start asking about concordance between workflows. So there's various ways to think about this. One, people have been talking about multi-analyst studies. Like, you can think of what we did as a multi-analyst study. One thing that's done in physics is, you actually have separate teams analyzing data independently without talking to one another, and often blindly. They don't actually know what's what. Things are just labeled ABC rather than, with actual labels. So they don't really know what they're looking for. Another thing you can think about is working with simulated data and making sure that all the analyses work when the data have the signals you expect. You can find those signals before you ever touch the real data. That's something we've started doing in my lab regularly now. At least for behavioral data. It's harder for fMRI data. Okay, let me stop for a second. This will be the next section. Let me just stop and see if there's any questions before we kind of move on to coding.
You mentioned kind of hyperparameter hacking, but in many tasks, so I work in clustering, and you'll see something like with the nothing, 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 and then with a set of hyperparameters, you get this nice picture. But if you know something about the way the algorithm works, then there's no reason to expect that it wouldn't work with the wrong hyperparameters. So how do you be honest with yourself, and how do you know, how do you tell them apart?
Clustering is really tricky, right? I mean, there's a great paper, whenever anybody in the lab says they want to do clustering, there's a paper I point them to by von Luxburg et al. Called Clustering, Science or Art, right? Because it is all of, like, there's no truth when it comes to clustering, right? There's a bunch of different, there's, almost an infinity of different results one can get. For me, the way that, what I want to see is you hold out part of the data, you design your clustering algorithm based on part of the data, and then you go ask whether on another part of the data that you haven't seen, do you get similarly reasonable results, right? And that will at least protect you from like overfitting to one particular sample from the data. Does that make sense? Other questions? Yeah.
So it seems very clear that the pipelines that you use can dramatically influence outcomes. But I guess going back to one of your first slides, when it was the meta-analysis of depression, just showing that there really wasn't any consistency when you looked at all the data. And I guess I'm wondering your thoughts on, like, with these higher like cognitive constructs that might have different phenotypes, or even like within the scans, people might have had coffee that morning or not had coffee, or they might be at different points in like hormone cycles and things like that. I guess what are your thoughts on like thinking about that, especially with regards to reproducibility prices?
Yeah, I think it's a great question. Clearly there are lots of, all the things you mentioned are all really important things. And if we don't measure them, then we don't know, right? Some of them are easier to measure than others. It's like most people are probably not going to want to give blood, so we can go do, hormonal analyses or something like that. But we can at least know about coffee. But I think and certainly there's some ways in which we can sort of try to control that. But in general, it's going to be difficult. And so the way that I think about it is, one, we have to, like, if we have big enough samples, at least some of that should wash out, right? So that's part of the solution. I think the other part is measuring the things that we know could be relevant, or controlling that. So, for example, time of day, right? we think that time of day probably has some sort of impact. We know that coffee has some sort of impact. So either controlling or measuring those things is probably a good idea. There was another hand. Yeah.
Are there like questions that you could answer with fMRI with like a couple of participants, like small, like you published on yourself, like
Yeah.
you had your N of 1,
Yeah.
right? It can't be the case that like you need 1,000 subjects for.
I'm gonna leave that to Katerina, who's gonna talk about exactly this. So
Cool.
yeah, no, the answer is yes, definitely. Now the one kind of set of questions that I think you just simply cannot answer without large samples are correlational questions, questions about associations between brain activity and something else, right, across people. That's just, that's the way correlations work. Yeah. Other questions? Yeah.
I have a question. Is there a recipe or a manual or an example that you follow for pre-registration? I looked up the pre-registration for MRI-QC that you showed, and it has these sections.
Yeah.
It looks like there's something systematic there, but
I don't feel like I have a great example. I mean, in part, it depends on, it's going to depend on exactly what you're doing, right? So, so for example, Michael here has done, a couple of registered reports around different types of fMRI analysis, but they're not really like generic, they're pretty specific to the particular use case. JB, do you have a comment?
I think that format is really journal dependent.
That's it.
First of all, there's few journals that deal with do the registration, registered report. I get registered report in reviewed registration. There are a few of those, and they have their formats, in their own formats. There's no, I don't know how to communication with that aspect.
Yeah.
It would be actually.
Yeah. So
especially for the case where you don't require data. Registered report and registration have been designed for when you are going to require data.
Right.
And many of those of our analysis now, just use existing data.
Right. I mean, I happen, there's actually a lot of, that's a great point. So, there's debate in the literature about whether it makes sense to pre-register data, studies of existing data sets. I think it's actually a really good idea, even if it's not as, clearly it's not as protective as pre-registrations for like newly collected data. But I don't some people think of pre-registration as like a way to prevent fraud or something like that. That's not how I think about it. I think about it as a way to prevent me from fooling myself, right? It's more about protecting me from like my, my biases and cognitive, I don't know what the word, biases. Yeah. And so I think it's really important. Other questions? So just to check time, do I have till 3? Is that right?
that's great.
Okay, cool.
Hello, sorry, I have a question.
Yep.
Thank you. So, so what I wanted to know, like, I work with EEG, with fMRI, but still is basically the same. It applies to both. When you were talking about the different ways of like analyzing the data and the different pipelines that you can end up having, for example, this is one of the things that have been the trickiest part, the most challenging part for me, because every time I look for a pipeline to try to use to analyze my data or to pre-process my data and then analyze it, I always found a different one applied. I asked 2 other scientists and they tell me, I am doing this, and then another one says, I'm doing this other thing. One said, I'm doing clustering. The other said, don't do clustering. So like so many different things, I end up getting confused. I don't end up knowing what to do. I don't want to end up doing, as you were saying, creating also my own pipeline and, continuing the same process that everyone doing their own pipeline. So what would you suggest in this case? Because I'm really in this moment of my research and I'm really curious about what to do. Thank you.
Yeah, no, thanks for the question. I mean, I think that it's a problem that lots of people are struggling with. I mean, you might think about it. There's this concept of the curse of dimensionality, right? Which has to do with like, the inability to model data as the number of dimensions goes up. But you can think of this as like another curse of dimensionality, right? The more high dimensional the data get, the more possible ways there are to analyze it. And so, we share those same problems with lots of other, omics analysis and stuff like that. So I guess the strategy that I would suggest is one, if you're only going to do one pipeline, make sure that's as boring a pipeline as possible and as kind of globally accepted a pipeline as possible. If you're not something, really kind of niche and sort of, not well established. I think it probably makes sense to make sure that your results are robust to the particular pipeline. And it's going to depend on the exact kind of questions that you're asking. So for example, there's a lot of debate in resting state analysis about whether you model global signal or not. There are going to be people in this room who probably disagree with this. I think that if you can't find a result both with and without global signal regression, then I would worry about that result unless there's a good argument for why exactly you shouldn't expect it to occur with or without global signal regression. So I would make sure that, especially for the things that might matter the most, that you're reasonably confident that your results are robust to that. Does that help?
Okay. So just to see if I understood, because one of the things that some of them suggested me was to, as you were saying, to have one pipeline or to have many different pipelines, to have like 3 different pipelines according to 3 different theoretical concepts that we have, and see if what you were saying, how robust the results are and which similarities I find between them as well. That's something that you were mentioning also from the fMRI when they were putting all together the results of all the analysis and see what similarities they have, but also the robustness as well they were mentioning. So I think that it's just like a little bit like still like discouraging in a way, because sometimes when you say that robustness of the results, it's like sometimes it's a bit tricky also what you find. So
no, I hear you and I feel for you. I feel for everybody who has to deal with these sort of questions, including the people in my lab. We don't have great answers for them, but it's, so at least thinking about them, I think, is important.
But thank you very much for that. I appreciate it.
Thanks.
I'll make it quick. When you talked about how there are different methodologies, and the slide had a bunch of fMRI results, I was wondering if all of that is post-analysis or if part of that is in the way that the fMRI machine is used to measure, because I'm not experienced.
Right. So for the one that I talked about with all the 70 different teams, the previous one, yes. So for like the depression meta-analysis, those were collected on different scanners, different protocols. There's lots of things that differ there.
Could there be a way to indicate what you used and then figure out how to like figure out the ambient noise and subtract it out individually? Depending,
people are trying to do that sort of thing. So there are now these large data sets like the ABCD study, which has data from 20 different sites or so. And each of those sites is a different scanner. Some are different brands of scanners, different types of scanners, and they differ quite a bit. And so people are developing tools to try to harmonize the data. Not clear that the tools that most people use actually work that well, but there's a lot of work going on trying to do this sort of harmonization. It's hard. And for most scanners, for most of those published studies, you don't have enough data from that scanner from different studies to be able to model that noise separately.
Right.
So you mentioned that your registration report, if you realize that analysis is not suitable, you can change it, and then you have to obviously mention it. So to what extent can you change your plan from the pre-registered report? And do you have to report the results from the initial analysis that you meant to do?
Can I just say it depends to all that? I mean, in short, I don't, I would think that there might, in one, it depends on what the reviewers want from you, right? Because it's going to depend who the reviewers are and how. It's also going to depend what particular changes you made, right? And they may, and what your justification is for the changes. And so they may well, want you to show the data done the way that was pre-registered and the way that wasn't. my guess is if you're changing, if you're changing a lot, people are going to start getting worried, right? But it also depends on the complexity of the study. If it's a super complex study with like, 1,000 choice points and you have to change things at 20 choice points, that's different from a study with 25 choice points and you change things at 20 choice points, right? So that's why I say it depends. I think it's hard to give a general answer, but I think it's going to depend on those types of concerns. All right, let's move on to coding. How many of you have written computer code in the course of your research? Okay, pretty much everybody in the room. How many of you have been trained in software engineering? Smaller number of hands. How many of you have ever written a test for your code? Okay, decent number of hands. So I think there are certainly clear examples of software errors being threats to reproducibility. The best known example is this person, Jeffrey Chang, who was and is still a kind of a hotshot structural biologist, had several papers in the early 2000s, 3 science papers about the structure of this particular protein, the ABC transporter, all of which had to be retracted because of an error in a piece of code. And my understanding is it's actually code they got from some other lab that basically caused a sign change. And that sign change basically changed the entire structure of the protein. Somebody else realized that. They ended up having to retract 5 papers. And because of that very tiny bug in the paper that caused the sign change, in the code that caused the sign change. And it's clear that, very small errors can have very big effects. I wrote a blog post about this a long time ago, where this was code that I had written to do. We were looking at open fMRI data and trying to do classification of cognitive tasks. So 23 people are doing one of 23 different tasks. And we want to say, can we use your fMRI data when we've never seen your brain before? Can we take data from other people and classify which of those 23 tasks you're doing? And I wrote what seems like, simple scikit-learn. Actually, I guess it was scikit-learn code. And I got 93% accuracy. If I got 100% accuracy, I would have known something was wrong, right? 93% accuracy seemed really good, maybe too good. So I had one of the people working in my lab, Sammy Koijo, look over the code, and he came back and said, "Oh, no, you screwed something up." Basically, I was missing the colon and the comma right here and right here, right? And so I was indexing the data backwards. And so when we did it correctly, we got 50% accuracy. Now, this is still much better than chance. Chance is 1 over 23 or something like that. But it's a lot less than 93%. And the paper was written when he found this. So we had to go back and change the paper. Fortunately, we found it out before the paper was submitted. So this shows how just a tiny, 4 characters can make a huge difference, right? We also have had this actually hit us after papers have been not published, but certainly put out on bioRxiv. So we had written a paper. This was behavioral analysis looking at the ABCD study and found some problems with the way they had implemented this particular task. And we reported that 1,300 of the 7,200 subjects showed this particular gross violation of a particular assumption. And so the preprint was out, the code were all available with the preprint, the data are all available from ABCD, and a member of the ABCD group dug into our code and found an error we'd made. This is, horrible looking piece of Boolean logic, and there was basically 1 parenthesis in the wrong place. And it basically caused us to fail to find some things. And so the actual number was 447 rather than 1,326. It didn't really change the conclusions of the paper. It maybe changed the severity of the conclusions a little bit. But, this was actually a great example, I thought, of, open code, open data, preprints, being able to actually save us from publishing this. This ended up getting published in eLife later that year or the next year. So it's an example one that, pretty much all of us, if we're writing enough code, are going to be making errors, right? There's no such thing as bug-free code. And the question is, how can you protect yourself from those errors as well as possible? And certainly peer review of this type, if you can get somebody like Sage Han to dig into your code, that's a great thing. Even if it's sort of a little bit adversarial, right? Because our paper was trashing on the ABCD study, at least on how they had implemented this one particular thing. It's a really amazing study, but we didn't like how they had done this particular set of things. But, the criticism drove them to really dig in, and it ultimately made everybody's science better, I think. Now, one big concern is what I call bug hacking, which is this idea that you're much more likely to find bugs if they disconfirm what you're looking for than if they confirm what you're looking for, right? If you like the results you get from the bug, then you're not going to ask yourself why is that happening? And so I think we have to figure out, ways to try to protect ourselves from that. And one is this thing that I call the principle of assumed error, which is the idea is like, if you find something that looks good, you should assume that it occurred due to an error and go try to find what that error was. And if you don't find an error after looking, a reasonable bit, if you're looking as hard as you would if the thing didn't work at all, then perhaps it's a real result. So how can we code for reproducibility? Obviously, you're going to learn a lot of stuff this week and next week about this, but I think that there are some principles that we can take. And I know you're going to have a talk about software engineering, I think, next week. And that talk will probably cover some of this. But I think there's some things that we want out of our code. We want it to be readable. We want it to be robust, meaning that it works when it should work and it fails gracefully and loudly when it should fail. We want it to be modular in the sense that you can reuse parts of it, you can test parts of it, and we want it to be well tested. And people, it can seem sometimes, I think, like, there are people who are just born being coders, right? And that, that it's like they somehow have this magic knowledge. Just like people think that, like, Jimi Hendrix is born knowing how to play guitar, right? Nobody's born knowing how to do anything except for crawl and babble, right? And obviously that's an overstatement, but there's all the stuff that we do that's amazing in the world is mostly stuff that we each had to learn hard. And what's actually, I think the thing that differentiates the Jimi Hendrixes of the world and like crappy guitarists like me is really the motivation to practice and play. And so the only way to get better at coding is to do it a lot and to do it in a way where you're actually trying to get better, not just like, doing it, but actually going and trying to learn, like, how do people do this stuff? Go watch videos, read books, right? It's just as if you were going to learn to play guitar, you're going to go read books about music theory and go watch videos about, whoever your favorite guitarists are. So just think about it as a craft, right? And everybody, anytime you start, I'm still a terrible woodworker. I don't do much woodworking. When I do it, it's really ugly because I don't do it enough to get better at it, right? So I think one really great place to start is coding style. I should say these slides are all available online. I put the link into the Slack channel for this talk. So feel free to take pictures if you want, but they are all available online. So I think that the first thing you can do is start basically coding with style, by which I mean coding in a way that's consistently organized and formatted. In part, this matters because, code, as Guido, the founder of Python, said, code is read much more often than it's written. So readability counts. And this, it's not just, other people who are going to be reading your code. You're going to be reading your code 6 months from now. And if it's not written well, you're going to have a hard time understanding what you were trying to do. Nearly every language has a convention for coding style. Python's is really good, I think. But following those is going to make your code easier for you to understand and for others. There's also kind of language independent principles of software design. Yeah, question?
Yeah, something I run into all the time is, especially if I'm on some kind of deadline and I don't yet know what will work and I don't know if I'm even doing it right. I'm going to write messy notebooks. And so at what point do I switch to writing it correctly? Because it would take too long, or is the idea to get good enough that it's fast?
Yes. And so there's a couple things to say. One is there's this quote by Donald Knuth, who's like one of the founding fathers of computer science, which is premature optimization is the root of all evil, right? So you certainly shouldn't be trying to do everything perfectly the first time around. Actually, this book, Refactoring, is all about what you said, right? The idea is you have code, refactoring is the idea of taking code and now making it better, making it more robust, better tested without actually changing its function, what it does, right? So I think it's, you know, you'll probably again hear more about this in the software engineering talk, but there's a lot of philosophies about how to design software, right? What people used to do, you know, 40 years ago, was to write down a huge document of requirements and say, these are all the things we want to do that the code has to do. And like figure out everything you want to do before you write any code. And then people realize that just doesn't work well. So now in the last 20 years, most, you know, software development groups have moved towards a philosophy that's called agile, where the idea is like basically build something that solves your problem, or at least that gives you kind of a minimum viable product that does something, and then iterate on it. Right. So rather than coming up with rather than like kind of thinking about all the things you want to do and then starting to write, which, you know, you'll never get to write anything, right? Start writing stuff, get something that works, and then the next step can be refactoring or, you know, sort of making it better. How to, you know, where that threshold should be, it sort of depends on what the code is for. I think, you know, if it's code that's going to be shared for like reuse, that's you know, it probably needs more work than if it's code that you're just going to be using for one project. I don't think there's any kind of, you know, general principles there, other than to say, I think it's better to write something and then with the intention that you're going to go back and at least, you know, somewhat improve it. Because it may, right, you might just like start and then give up, and you don't want to put a ton of time into having a perfectly crafted thing that you're going to then throw away.
So I guess, when do I know I should go back and put as much into the refactoring as I'm planning to do before moving on?
Yeah, that's a tough question. I don't know. Does anybody else who builds software in the room, does one of you want to say how you think about that? Yeah.
I guess I'll just say that I feel like it's almost like a continuous cycle. Like you write, you get code out there first, and then you use it enough to know where the pain points are and what's wrong, and what parts aren't working very well and what parts are really poorly explained, and you struggle to go back at it, and then you refactor, and you either you don't start from scratch because you understand what you're doing this time, but you maybe rewrite a lot of chunks of it and reorganize code with the code and add more comments, and then you start over.
Right. So Fowler came up with this idea called a code smell, which is basically like I don't know if I have a slide on this, like things that you see when you're looking at code that aren't like errors, but they just smell bad. Because you know that they're going to be brittle, or they're going to be hard to maintain or whatever. And those are the kind of things you want to target. Maybe the one other thing, the one other signal I think for maybe it's time to refactor is like if you're trying to add something in it and it turns out to just be really confusing about how to do stuff, right? If the code has become so sort of, you know, unwieldy or unmaintainable or unextendable, that you're like having to like get really, it makes you anxious to think about adding a little feature. That's a signal that maybe you should think about refactoring.
But at that point, it has now become hard to go back and refactor because I can no longer figure out what everything did.
Yeah, so let's come back to that in a minute. We're going to come to another way to deal with that problem. That's a yes. Okay, but first, I'll only say one more thing. So books. There's a lot of books out there, some of which are like some of these pragmatic programming and clean code, I think are both like 900 pages. Actually, pragmatic programmer is really good, but it's long. If you had to pick up one book, the book I would pick up is The Art of Readable Code. You can actually find a version of it, a PDF of it online for free if you want it. It's a really great book about how to write clean code. It's kind of language independent. It uses examples from various languages. So you have to be at least comfortable. I mean, just if it's from a language you don't understand, just gloss over it. That's because it's really about, you know, the principles that they're talking about. One of the things that I think is commonly thought amongst people who haven't read much of the software engineering literature is that the thing that distinguishes good code is lots of comments. At least some people in at least in the sort of clean code movement would seriously beg to differ. So that there's a couple of quotes at the bottom, right? Clear and expressive code with few comments is far superior to cluttered and complex code with lots of comments. Or don't comment bad code, rewrite it. So the idea is that like your code should really be understandable without any comments. If you've written things, if the functions are named in ways that are readable, the variables are named in ways that are readable. If it's readable code, it often doesn't need any comments, except as what people sometimes talk about, like the director's commentary, right? Things about intention. Why are you doing this thing, right? Or, you know, if there's a particular weird, you know, edge case that you're working around, why, you know, why are you doing this thing that doesn't really make sense? Things like that. In general, the code should kind of speak for itself. So if you're in the Python world, then there's a lot of tools that can help you write better code. Things like static analysis. So there's tools like, I use Flake8, they're called linters in general, that basically can take a piece of code and tell you what's wrong with the code. Where, you know, one, does it have any errors? So before you go run it, you can look at it and say, hey, are there any actual errors in the code, like syntax errors? And also tell you, like, where are you not following the style guidelines? There's also tools that can take your code and reformat it to make it follow the style guidelines, like black or blue or two of those things. I don't use those extensively. I'll use them occasionally if I'm like lazy and write some code that I haven't, where I've kind of not done a great job with standards compliance, then I can just run it through and get it fixed. You know, what you should probably do is, you know, whatever editor you're using, like I use VS Code. I'm kind of a advocate for VS Code because it has a lot of good integrations, but one of the good integrations is Flake8. I mean, most editors now have Flake8 integration, but it'll basically, every time you save it, it'll just pop up with a window that says, hey, these are the things that are wrong with that code. So code review, I think, is a really important thing, which basically means just, you know, having other people looking at your code. And this can work in a lot of different ways. I mentioned Linus's law, I think before, right? Given enough eyeballs, all bugs are shallow. There's, you know, in my lab, we regularly will do code reviews where, you know, one of the people in the lab puts up their code on the board and, or on the projector, and it's not like saying, hey, you screwed this up. It's really saying, why are you doing this? What's the intention here? Why was this written in this particular way? And I think I think it actually has less benefit for the person whose code is being reviewed than it does for the group in general, kind of, you know, learning how to look at code, how to think about code and make it better. This paper by Petrie and Wilson, I think, lays out a nice set of ideas for how to do kind of scientific code review. Greg Wilson in general, I think, has written a lot of good stuff about kind of code quality. I'll talk tomorrow a bit more about AI tools and their utility for code review. I think that they're certainly not as good as humans in a lot of ways, but they become really useful. I think, especially with GPT-4, has become really useful for looking at code and telling you things that it sees in the code. But again, I'll say more about that tomorrow. So testing, is there gonna be any sessions on testing? Okay, so you'll learn about testing. I mean, testing is kind of it's a very complex subject, in part because, you know, there's lots of kinds of tests one can do. A lot of them, I think, aren't necessarily that useful for us as scientists, but there's a number of things you can do that are really important. One is like just sanity checking, making sure that all the values are in ranges that make sense. If you're writing functions that are supposed to do a thing, then it's probably a good idea to have a test to make sure the function does that thing correctly. Here's an example where testing could have saved somebody a bit of a headache. Paper published in a few years ago in PLOS One that had made some claims about conspiracy ideation and science rejection. And it turns out the data set included two notable age outliers, reported ages 5 and 32757. And that when you take those out, it changes the results. Surprise. If they had just said, hey, let's make sure that ages in the data set are within the age range that the study was supposed to have. And this single assertion would have caught that and made sure that, you know, that they didn't do that. Similarly, like if you're measuring reaction times, checking to make sure the reaction times aren't negative is probably a good idea. So to get back to this question that was asked earlier about, you know, I want to refactor my code, but I don't know how to make sure that it's doing the right thing. And so there's an idea called test-driven development that I find I've started writing more and more code using this sort of idiom because I find it really helpful for myself to think about what I'm doing. The idea is that before you write any code to actually solve your problem, you write tests that will pass when you've solved the problem. When your code does what it's supposed to do, the test will pass. So you start writing code, the test is going to fail. Once your code passes the test, you're done. Until you decide, oh, there's another function that I need. And then you write another test for the other function and you start adding that functionality. When you pass that test, you're done. So the idea is that any requirements of the code are defined by the test, right? So what you could do if you have a code base, right, that does some things, presumably there are things that you expect the code base to do. You could write a set of tests that basically make sure that the code does the things that the current code is supposed to do. And then you can start refactoring to basically fill in, to make sure that the new code passes the tests in the same way that the old code did. Right. So it's the tests become like an objective function, like a criterion for saying, hey, the code is working. And I find this to be a powerful way to think about if I'm starting to write code, especially, I mean, if I'm just like doing a little data analysis thing or something like that, maybe not, but if I'm doing something more substantial, I'll often start out thinking about like, one thing it does is it makes you think about what's the problem I'm trying to solve, right? From a slightly longer view. And then often you'll realize, oh, it passes the test. You're like, oh, that wasn't really the problem I wanted to solve. It's a slightly different problem, and then I can write a different test for that. The other cool thing is that then you can make sure that if you go back and add code later, it's not going to break the functionality that you put in before, right? We call that regression testing. Make sure that you haven't like broken something that you wouldn't that might not be obvious now, but if you then went back and tried to do it later, that you would see that it's broken.
Another point about testing. So is it like the same as asking, basically say if the test, if there is any mistake, just spell the error, or is it not? Is it like, and the other way to
That's a great question. So there's lots of different types of testing. In general, the kind of testing that I'm talking about would be testing of, let's say that you have I'll show an example tomorrow in my AI talk of, I want to write a function that will tell me the escape velocity of a planet based on the radius and mass or whatever it is that you need to know of that planet, right? There are things you need to know to compute how fast do I have to go to get away from the planet. I can write a function to do that, right? And then what I want to do in my test is basically say, well, if I know, for example, for Mars, I know that its mass is this and its radius is this and its escape velocity is this. I make sure that when I give my function these inputs, that it gives me the correct output, right? So it's not about does it crash, it's about does it behave correctly. Similarly, like if I give it a negative mass, it should say, hey, wait, you can't do that, right? It shouldn't go do some sort of, it should give me, and when I basically, if I give it garbage, it should tell me, no, I can't work with garbage, right? If I give it a usable input, it should give me the correct output. So those are the kind of things that the kind of testing that I'm thinking about would do. Does that help? Cool. All right, now there's a ton of tools available for sort of automated testing. If you work in GitHub, GitHub has this thing called Actions, where you can make it so that every time you check in, every time you commit some code to your GitHub repository, it can go and automatically run the test to make sure you haven't broken anything. And that's a super useful thing to do. So, sorry, you can't read this. This is basically just like what it'll help you automatically generate for a Python application. It'll help you automatically generate this particular workflow file that you need to do the test. The code has to be kind of ready to run with PyTest. You have to have built the tests, but then it'll go run the test. And in this case, the test crashed. That's why it has a big red X up there. And now I need to go fix something. Ironically, it's from my PyTest tutorial for which the tests are now crashing because I broke something. So I just want to end by just sort of mentioning that, I focused a lot on reproducibility, right? But something can be reproducibly wrong, right? Reproducibility and validity are not the same thing. In psychology, we talk about reliability versus validity. And the idea is like, you want things to be both reliable, which is they consistently give you a very similar answer, and valid in the sense that they consistently give you the right answer, right? Something can be really reliable, like really, not have much noise in it, but give you the wrong answer. And so I think we need to think about how do you validate your code. And so I've already mentioned a little bit about simulation, but I think that, your code should be able to basically give you back, if you're trying to estimate something about the data, you should be able to put in simulated data and have your code give you back the answers that it should give you, given the known signals that are in the simulated data. Similarly, if you give it data that are just noise, it should basically come back and say, hey, there's nothing significant here, or at least, there's only 5% of significant things if your alpha is 5%. There's a lot of tools for generating simulated data. I played a bit with the synthetic data vault, which seems pretty cool. There's a lot of other tools for, we still don't have great tools for like full-blown fMRI data. I wish we did. There are some tools out there that just none of them are kind of like well featured enough to do everything we need. But for a lot of analyses, you can come up with simulated data. And so what I now do, again, primarily for behavioral analyses is, I will have students basically, often like if we have real data, sometimes I will for one of my students for their first year project, I took the real data, I generated synthetic data based on the real data and gave it to them and said, hey, go analyze this and tell me what the answers are. And once they came back and told me what the answers were and they got it right, then I let them analyze the real data after they had sort of checked that in and actually pre-registered the code. So that's another way to just make sure that your code isn't just reproducible, but it's also getting the right answer. Okay, so reproducibility problem still around, unfortunately, but I think there's lots of tools. We know a lot more about it and how to fix it, and you're going to learn a lot about how to fix it and go on and do stuff that'll prevent me from having to give this talk 10 years from now, hopefully. So I just want to thank everybody in my group, our collaborators, and thanks for inviting me. All right. Yeah.
You referred to your blog previously, and I tried to Google it, and it seems that your website has changed. And is there any way we could like find those blog posts?
Oh, yeah. Sorry about that. If you go, yeah, I'll put it in the Slack. Yeah. They all exist. They're just, you have to like take an extra step. Yeah.
I hope I'm not the only one here, but so in my experience, good code is not exactly encouraged in academia. Like I've been asked to get things done quickly, or like that's never been a priority. And taking the time to try to learn those principles, I've actually gotten feedback like, don't be doing that. We need you to do the work. And then everyone's code tends to be like crazy. So what is a good way to, I guess, work with that and like make myself do it anyway, or
yeah,
I don't know.
Yeah. You're definitely not alone. I'm sure that there's many people in this room who either have or will experience exactly that bind. And I don't know a great answer, right? I live in a bubble where everybody wants to do this stuff and is encouraged to do it, but I realize that a lot of the world extends beyond that bubble, right? And I hear stories all the time about people getting negative feedback from their supervisors about all the stuff that I talked about, pre-registration, spending time on coding, trying to do anything that slows research down, right? And I think that probably shows in their work, right? They're probably in the little red bubbles in the figure. And if it doesn't, it will probably at some point, because that's just not conducive to good science. Unfortunately, the incentive structures are push, exactly towards that type of work. So there's work going on to try to change that. At least in the US, there's a group that I'm involved with called Helios, Higher Education Leadership Initiative and Open Scholarship, that's basically working with, it's like 80, 90 universities now working together to try to change incentives to incentivize reproducible and open science. But it's really hard, you know, it's like a really big ship. It turns very slowly. And the problem, you know, another problem is that there's a lot of people of my vintage and older, you know, who got their careers doing all the stuff that I'm now saying is really bad, right? Including me. I mean, there's a lot of things that I did 20 years ago that I cringe at now, right? But I'm, you know, I'm okay with that because I think science is about getting better. A lot of people take this as you know, as a direct criticism of them, and are not comfortable with that. And yeah, it's just really hard to know how to deal with, like, you know, old people who like are going to be like that. I don't know how, you know, other than, I mean, I go give these talks everywhere. I try to, you know, be an example. A number of other people in my cohort are trying to be examples. It's really hard to know what you know, how to change. I mean, I wrote a book about how behavior change is really hard. Behavior change is really hard. And this is an example of where, you know, PIs who grew up in a different world need to change their behavior. And, you know, a lot of them are probably just like hoping that they can retire before they have to, you know, sort of actually think about the stuff, unfortunately. I mean, you know, that's just the way it is. So I'm sorry to have such a depressing answer. I just, you know, it's behavior change is really hard. Yeah.
Yeah, going back to like now that you know that this is like a bad thing, wouldn't it make sense to fund studies that go back and visit like classical studies in new science and just take a look at whether they're like valid? I know there's like some efforts being put to it. Is it like something, for example, that is encouraged by the NIH to like give you money to go and like check studies that people use kind of involved?
Encouraged, I don't know. I mean, you know, NIH certainly, you know, has rigor and reproducibility as major components of their review process now, at least in terms of what the reviewers are told when they see a grant proposal. But ultimately it comes down, you know, how does grant review work, right? It's a bunch of people like me in a room who have a big pile of proposals, and they have to say, hey, how do we like what this one says? Is it interesting? Is it innovative? Blah, blah, blah. Is it rigorous and reproducible, right? It totally depends on who's in the room. Like if I'm in the room, I'm going to make noise about those issues. If, you know, there's another random sample of people who would not care about that stuff at all. So there's a sampling variability just about, you know, how the review is going to go. But I think that there's, you know, my impression is that the people at NIH, the people who work in the programs at NIH are really care a lot about this stuff. I mean, they're giving money out to people to go do science. They want it to be real. Like you can imagine how they felt when they saw that, you know, their darling 2017. I didn't show you that NIH had a press release about the 2017 suicide study back in 2017. You know, clearly they were not happy when they saw that they'd given all this money based on basically a piece of problematic science. They really want to get the right answers, in part because Congress expects them to put their money behind getting the right answers, and also because it's, you know, they want to solve medical problems. They want to figure out, you know, how do we understand what's going on in depression and, you know, possibly fix it. And that's not going to happen if you're basing it on, you know, sort of science that's not reproducible. So I think they care. It's just it really comes down to convincing the reviewers to care.
I don't know if today or tomorrow, would you have time to like tell us about how I see fMRI prep as like a big success story in like this movement, because like people have really adopted it and people use it. Like what makes a tool something that people are going to use and adopt and like it's going to be well maintained versus like so much of the stuff I see is just like it was on GitHub and posted 3 years ago and no one ever touched it again. Yeah, and it had all this potential. Yeah, I'm happy to have a chat. I mean, in short, it's really hard work to maintain software and not very glamorous, right? In the academic sphere too, because like, what's the, like, I could get a software job out of industry.
Yeah.
10 times the money.
Yeah, yeah.
Have the resources people to help.
Yeah.
I just don't understand how it's been possible that you did that.
Yeah, I mean, I have 4 software developers in my group that have, some of which have been in my group for, 5, 6, 7 years. I think they like it there. There are people, so, one thing I can say is like, everybody in my lab knows they could go make twice as much money in industry. They don't want that, right? They want to be, one, working in an environment that is fun and also working on problems that they think matter and not have to live with the toxicity. Most of them have worked in industry
Do you
before.
get like an RO1 to fund fMRI prep?
We have an RO1 like grant. Yeah. So the Brain Initiative has grants that fund software development. So we actually have a grant to fund that. We have another one funding open neuro. Yeah. So there are grants available, particularly through the Brain Initiative for software development. The Chan Zuckerberg Initiative has also been funding. Did you get for DIPI? Yeah. So they fund NILEARN. They funded a number of, those are smaller grants than an NIH grant, but nonetheless, it's, decent money for a couple of years. So there's a little bit of money out there. It's still hard. Like I wouldn't, if I was, it's different. Once I had tenure, it doesn't matter what I do, right? I mean, it matters, but not that much. As long as it's kind of, interesting science. But I don't know that I would have focused so much my efforts on like software development, in part because, the fact that fMRI prep succeeded is, we're very happy about it, but there's almost certainly some randomness to why it succeeded, right? I think it succeeded in part because it was good software, but also probably just happened to be at the right time, the right place, the right people got behind it. Chris Gorgolewski was really good at convincing people to jump on the bandwagon early. It's a gamble as to even a really good software package might go nowhere just for kind of random reasons, just like really good Rick Rubin, the music producer, has a book. I heard him on a podcast. He's talking about how he finds it really impossible to tell whether an album, there might be albums that he thinks are just amazing and are going to be super hits and they'd never go anywhere. And it has to do with what else is going on the week it was released, or, what other things are going on in the world. So it's really hard to predict how things are going to succeed. And so maybe another way to say it is you don't want to overfit too much to the things that have been successful.
Okay, let's thank Russ again. Right until 3:30 Pacific time when we're here in all the